This presentation will cover the tests that are done to help identify gram-positive organisms. So when you set up your cultures, and we're looking particularly at organisms that grow within an aerobic environment, which is growth at ambient air, 35 to 37 degrees Celsius. So with a routine culture of a blood agar and MacConkey, then your gram-positive organisms will grow on the blood agar but not the MacConkey plate. So your MacConkey is selective and differential for your gram-negatives. So if we see that there's no growth on MacConkey, growth on blood agar, then we can probably assume that it's going to be a gram-positive organism. So again, our gram-positive organisms are divided into gram-positive cocci and bacilli. So first we're going to look at the gram-positive cocci. Our first test would be our catalase test. Once we determine if it's positive or negative, then from there we would determine what test we would do for each. So based on appearance and catalase positive, we would look at our Staphylococcus or Micrococcus. And then from there we would differentiate those out depending on which genus. And for catalase negative, Streptococcus and Enterococcus. And depending on how it appears on the plate will depend on what particular test we do next. So these organisms typically grow on sheep blood agar, but they can also grow on other types of media. So here we have a list of media that would be used to help with growth and identification of our catalase positive gram-positive cocci, and then also our catalase negative gram-positive cocci. So with our catalase test, what happens here is that a little bit of the organism is placed onto a slide. And so you would take a stick or a culture loop, place that organism onto the slide, and kind of spread it out a little bit. And then from there you would drop hydrogen peroxide on that organism. And if the organism produces bubbles, and so what happens here is the catalase enzyme, if it's present in that organism, will convert that hydrogen peroxide to water and oxygen. And then as a result, you will see that oxygen to start to bubble up. So here at the top, you have a positive reaction for catalase, and at the bottom, you have a negative reaction for catalase. So you do not see any bubbling. And one thing that can interfere with this test is that blood will actually give a positive test. So if you've ever gotten blood on article of clothing or anything and you've added a drop of peroxide to it, or even on a wound, you'll see that there are a lot of bubbles that are created. And so you want to avoid getting any of that blood agar accidentally from that plate, because that blood agar could give you a false result. So once we've determined our catalase reaction, then from there, if it's positive, we're going to determine if we're dealing with a staph species or a micrococcus. So with our staph, the first thing we're going to do is, if we suspect a staphylococcus, then we're going to do a coagulase test. So there are two different types. Our first test would be a slide coagulase, in which we would take some of that organism and mix it with the reagent, and what we're looking for is clumping. So if the organism is positive, you will see clumping visually with the eye within about 10 seconds. And so you can see here at the top where there is a positive reaction, and then at the bottom there is a negative reaction, so no clumping. And so a positive would indicate a coagulase positive staph or a Staph aureus. If it's negative, then you may be dealing with a coag negative staph. If the coagulase test is performed in a tube, then this would be indicated by a clot would be positive. So you can see at the top where you actually have like a gel type clot. Sometimes they may be a little bit smaller, so you would have to rotate that tube to see it. And then at the bottom there is no clot formation, so that will be considered negative. You also have the modified oxidase test. And with this, you're looking for a positive result of a blue to purplish color, as demonstrated at the top, or no color. And don't mistake the actual drop of the reagent that kind of puts a tinge on the disc as any sort of color change, but you're looking for no bluish purple color for a negative reaction. Bacitracin susceptibility. With this particular test, you would perform an inoculum of the organism onto the culture plate, and so you would make sure that this organism creates a blanket all over this plate with a lot of growth. So when you culture it, you would use typically a broth suspension, maybe a swab, and you would rub that almost like you were coloring or something onto that plate or an area of that plate. Once you've inoculated it, you would place a disc, a paper disc that has the antibiotic impregnated inside of that disc onto the plate. You would incubate this plate overnight or 18-24 hours to allow that organism to grow. And what will happen is that the antibiotic from this disc will leach out into this agar. And as it leaches out into the agar, if the organism is susceptible, then it will not grow up to a certain point where that antibiotic starts to dilute out. And if it does not grow, it's considered inhibited. And so if that growth is inhibited, and when you measure this from one side to the next, if it is greater than 10 millimeters, then this is considered susceptible or sensitive to this particular antibiotic. If, however, it is resistant, then there is no zone of inhibition, and that organism will grow all the way up to that disc. Novabicin is done the same way. You have that disc, that NB, that is impregnated with that organism. If there's no growth or a zone of inhibition within greater than 16 micrometers, so from one side to the next of that growth pattern. And these little marks here are just the culture streaks. That's not actual growth, so you can see the more cloudy edges where that organism is actually growing. And so if you measure from one side to the other with that growth pattern, greater than 16 millimeters is considered susceptible or sensitive. And then if you have growth all the way up to the disc, then that is resistant. If you're checking for methicillin resistance, there are a couple of different test methods that are used. Using the disc similar to the last couple of tests, you can use the oxacillin disc to check for that methicillin resistant. So you can see that disc here up top. Or you can also use the cefoxitin disc here at the bottom. And what you're looking for is an organism that is resistant, so you have growth all the way up to it, and an organism that is sensitive. And so if that organism is resistant to that particular disc, then it is considered to be resistant to methicillin. And so this would be where you would determine if this organism is MRSA, or methicillin resistant Staph aureus. And so in that case, most often this organism can be treated with vancomycin, although there are some Staph aureus species that have started to develop a resistance to vancomycin as well. So next we're going to take a look at our catalase negative gram-positive cocci tests that we do to differentiate these organisms. You can see here in this flowchart, first thing we're going to do is base it off of appearance. And so with that, what we would do is observe the hemolysis pattern within our culture on our blood agar plate. And so a lot of times with throat cultures or genital cultures, the technician will do what we call is a blood agar stab. And so when you're setting up this culture, when you do that streaking technique, and several of those streaks, you will take your culture loop and actually stab the agar deep down into that agar several times and in several different spots. And so what happens here is that it allows for the detection of streptolysin O. And streptolysin O is an oxygen labile virulent factor that is produced by Strep pyogenes. And so with that, the streptolysin O works best when it's not in the presence of oxygen. So when you stab that agar plate, it gives it an anaerobic type environment to grow in, and it enhances that hemolysis pattern. And so again, just to go over the different types of hemolysis, you have alpha hemolysis, which is a partial lysin, and you have a greening type hemolysis. Beta hemolysis is a complete hemolysis, and you get a complete clearing or a yellowing type color. And you have gamma hemolysis, which is no hemolysis. The PYR test is indicated as a positive test if you see a bright red color, so that organism is added to that disc, so you can see the outline of that organism here. And then a reagent is added to it, and it's allowed to sit for 5 minutes. And you look for that pink-red color for positive, and then no color or an orange type color for negative. So you can kind of see a little bit where that culture might be here. And then that reagent is added. So do not mistake that little bit of tinge from that organism and that reagent to be a positive. So for our hypurate test, we're looking for that deep purple color to be a positive, and then no color or a slightly yellow tinge would be negative. So with our CAMP test, what you do is you take an organism that has a beta lysin in it, and so with this would be our organism would be something like Staph aureus. You streak that organism down the center of the plate, and then our organism that we're testing for the CAMP test, we would streak it perpendicular up against that Staph aureus streak. And then after it has been streaked, you will incubate it. And what you're looking for is an enhancement to that hemolysis. And so what it will do will create a bit of what we call a arrow or a campfire type appearance, where it kind of expands out with that hemolysis. And then a negative, you would not see that enhancement of that hemolysis. So this would be considered positive, and this is negative. With our bowel esculin test, we're trying to determine if the organism can hydrolyze esculin. And so with this, what you will do is you will actually take one of your colonies and you will inoculate the slant of the agar. And so once you inoculate it, you allow this organism time to grow. And then what you're looking for is the blackening of that agar as that organism will hydrolyze that esculin. And if there is no growth and no blackening of that agar, that is considered a negative reaction. Blackening is positive. Bowel solubility is used for your strep pneumoniae. And so what you will do is if you suspect that you have an alpha hemolytic streptococci and you want to determine if the colony itself is lysed by the bile or the bile salts, then you would add a small drop to the agar plate. And where you see here that you've actually got nice little colonies, and then that reagent has been dropped onto that colony plate, you can see the remnant of a few colonies here where those colonies have been lysed by that reagent. And so as that colony disintegrates, it can start to kind of leave an imprint of where that colony was at. And so that would indicate a positive test. And then of course if you were to drop it on the colony and the colony remained, then that's considered negative. And so these two here together side by side, you don't have a negative on the plate. Down here it's just where the reagent has not come in contact with those colonies. Your salt tolerance test, or your 6.5% sodium chloride. If you have an organism that is resistant to a high salt concentration, so an example of this would be your Enterococcus, then what you would do is, if you're suspecting that, you could inoculate a broth media with that colony, and then you allow time for that organism to grow. And what will happen is, as that organism is able to grow in that salt media, then it will start to turn cloudy, and if there's an indicator within that media, then that indicator will change from purple to yellow. And so this would be considered a positive test. It's really hard to tell that it's cloudy, but it is cloudy. And then this one is negative, there's no turbidity, and that indicator has not changed. Our Optogen or P-disc is set up just like our other disc test that we did. You create that bed of that organism, place that disc on it, allow it time to grow, and then you're measuring that growth pattern from one side to the next. Greater than or equal to 14 millimeters is considered susceptible or sensitive. And then if it grows all the way up to it, it is resistant. So one of the other testing we can do for our strep species is to do Landsfield grouping. And so with Landsfield grouping, this will classify these organisms based on the type of carbohydrate that is present in the cell wall. And so you will have several different reagents with these kits. And so here you can see some of those reagents. We have A, B, C, D, and F. And you will use these little cards here to test each of those reagents. So if you were to be testing A and B Landsfield groups, you would take a little bit of that organism, you would put it on to that slide. You will mix a drop of that reagent, so the A reagent, B reagent. You would mix them together, rotate them, and what you're looking for is clumping, which will usually kind of float out around the edges here. And you can see a little bit of clumping here inside of this reagent, whereas your negative would kind of be a smoky type appearance with no clumping. And so with your positive result, so for in this example, if it was positive to our A but negative to our B, then we did the Landsfield typing here for our group A S. pyogenes. So Strep pneumo and viridans streptococci do not belong to any Landsfield group, so this test would not be useful for those. And what you're looking for is, once you kind of look at your hemolytic pattern and you're trying to decide for sure if it is one of these particular organisms, then you would test it against this reagent to help narrow down what organism you're working with. So when performing Landsfield grouping, primarily what we're going to discuss will be the ones that we are covering, which is Group A, Group B, Group D, but also you need to know that your Enterococcus was once referred to as a Group D strep. However, it's since been classified into its own separate group. So if you were to get a positive for a Group D, you would still need to further differentiate it into an Enterococcus or another Group D strep or a non-Enterococcus Group D strep. And then, of course, your strep pneumoniae and Viridin strep do not fall into a grouping. So those would be diagnosed or identified based on other characteristics. Next, we're going to look at our gram-positive bacilli and the tests that we do associated with these organisms. And again, we divide them out to spore-forming and non-spore-forming. And then from there, based on what particular organism we're dealing with, will depend on what type of testing we do to determine that particular organism. Again, here we have our charts. Just as a reminder, we are not going to be talking about these organisms here. Those will come later. With our motility test, we are looking either in a direct wet mount, where we incubate that organism in a broth type media, and then we look at it for that end-to-end tumbling motility for our Listeria after 1 to 2 hours of incubation at room temp. Or we can use an agar. So with that agar, we would take that to culture onto a needle, which is just basically a piece of straight metal. And then we stab that down into that agar. And you can see here where that organism has grown all along that stab. And then what we're looking for is if it has motility, then that organism is able to diffuse throughout that agar. So you can see here at the top where you have growth within that agar outside of that slant. You can see the growth coming out here. So the whole agar kind of appears as a cloudiness, whereas this one is clear around those edges outside of that stab. And then of course here you have that umbrella motility from that Listeria. So you can see where it kind of forms that little circular type appearance here up top, and then you have that stab mark that comes down and creates that little umbrella look to that organism. So our esculin hydrolysis is very similar to the bile esculin test. The main difference is the tube that is used within your bile esculin is a selective type medium. So they're essentially the same as far as how the organisms present, which is that blackening of the agar. So you can see that here would be positive, and then here we have negative. The triple sugar iron agar, it's also used for your gram-negative organisms, but it can be used for our gram-positive bacilli when we're looking to see if it is producing that H2S. And so what happens here is the organism is inoculated again with one of those straight metal needles, and you would stab down into the agar, and what you're looking for is that blackening within that agar in which it has produced that H2S. With our methyl red, we're looking for the red color, which would indicate a positive test, or a yellow color, which is negative. So this is our MR. And then here we have our VP. And so with this one, you're looking for this red color. And then this one, of course, is negative. So with the oxidase test, the organism is put onto the filter paper. So it's allowed to sit for a few seconds, and then you drop the oxidase reagent onto that organism. And what you're looking for is this deep purple color, which would indicate a positive result. And then here at the bottom, you see no color, so this will be a negative result. So there are a couple of different medias that can be used for culturing and differentiating the Bacillus species. And for example, with your bicarbonate agar, if you're suspecting anthrax and you put it on this agar, then it's going to induce a capsule formation. And so what will happen is if that capsule forms, then you'll get this real mucoid look. You can kind of see a thickening here, whereas these outside edges look kind of watery or runny. Down here is a good example of that watery look. And so with this, you could suspect, if you get this appearance, that you're dealing with the anthrax, whereas this is definitely not anthrax. You don't see that mucoid look. You can see the difference of the appearance on the blood agar over here for both of these. So you can still see that kind of straightforward look here. And then you also have a couple of other medias that can help to differentiate these organisms. With your Corynebacterium, you could use these two different types of media here. So like your TIN, your Tansdale media, where your organisms would give that black appearance or that black halo appearance when it's cultured onto those agar plates. And then you have your serological testing, in which you're looking for those different toxins where that organism is streaked onto that plate, and then you have a antitoxin strip that's laid onto it. And depending on that bacterial growth, so you have your bacteria growth here, so you'd streak a line across, and you would do that several times, lay that strip down, and what you're looking for is these little precipitation lines to help you determine the particular toxin.